Hello and welcome everyone to our file merging webinar today. My name is Rafael Gomez Amaro. I am a technical application specialist at Novo Software. In case you are not familiar with us, Novo Software has been providing flow and image cytometry data analysis software for over 20 years. Our headquarters is located in sunny Glendale, California, and we have employees spread out all across the US and around the world. And if you're just joining into us here, if you have any questions, feel free to type those in throughout the webinar and our staff will be answering them. You'll find the questions box here in the GoToWebinar little window. You can expand that out just by clicking on this orange button. So today we're going to be talking about the new file merging features added to FCS Express 6 in our latest update. In this release, we have added the ability to virtually merge multiple data files on the fly using the new Merge FCS Files option. With the Merge FCS Files option, you can now merge multiple data files directly from the data list and plot them just like you would for any single data file. Merging data files using the new Merge FCS Files option is a convenient way to quickly load and visualize multiple files on one plot and can really be helpful when you are comparing or identifying changes across samples. So why did we add this feature to the software? Well, usually with flow and image cytometry files in FCS Express, files are added as separate data to our analysis. And if we wanted to merge them, we would need to use our batch export dialog to merge and concatenate those files together. This is a flexible multi-step process, but it requires you to save a new merged file. With the new virtual file merging option, we can now simply merge our data files much faster and more conveniently since it's all done from the data list. And there's no need to export the result as a new data file since the files that you have selected will essentially be virtually merged on the fly and just held in memory for you to work with in your layouts. In addition, the new merged files can also be represented in a heat map, even if they were not acquired on a plate. So with the latest update to version 6, we now have two options for file merging. The traditional batch export method, which still provides the most flexibility for file merging, since your files can come from any folder, use any compensation, and you can even select down to the gate which events will be exported and merged. However, you still need to save that merged file as a new file. Or you can use our new Merge FCS file option, which we'll be talking about today. This is for when we want to quickly merge and view all of our data, but may or may not have a need to save that merged file for later use. While less flexible, virtual file merging is extremely fast and convenient. Now, when should we use the new Merge FCS file option or virtual file merging? Well, it'll be particularly useful when we want to quickly combine multiple samples and visualize them together on a single plot. Like, for example, when plotting an antibody titration or drug treatment series. We can also use file merging for performing things like data transformations on multiple files simultaneously without ending up with different transformation results for each file. So let's say you want to perform something like tSNE on biological replicates across experiments or drug treatments. Merging your files will result in a more complete and representative data set for all of your samples, and that can really help to improve your results. With virtual file merging, we have also created this new virtual plate. It's a brand new option that we've introduced with the Merge FCS file that lets you quickly visualize and compare your merged data on a heat map, even if that data was not acquired on a plate. So in the next 25 minutes or so, we're going to go walk through several examples using the new virtual file merging option, this merged FCS file. First, we'll go over a simple example and learn the basics of the Merge FCS file option. Next, we'll show you an example layout with titration data, where we'll show you how easy it is to use the Merge FCS files with templates and how we can use a virtual plate to just quickly visualize changes across our data set, compare multiple samples with each other, all on a single plot. And finally, we'll cover an example using data transformations and tSNE, and we'll show you how file merging gives us a more flexible analysis. So let's go ahead and get started with merging some data files. In this first example here, we're just going to start from scratch with a new layout. So the first thing I'm going to need to do is just open up a new layout here. So I'll just go to New and create my new blank layout. And what we're going to do is just start from the data list. And that's where file merging is going to be happening, at data loading using the data list. So I'll go to my data tab here, and I will open up my data list. And the first thing we'll need to do is merge our files. So I'll go and add some data files to this analysis here. So I'm just going to navigate to where my files are, and they're going to be in this folder here. And you'll see that I have multiple FCS files here. And what we're going to do is we're going to select the new Merge FCS Files option to merge these files. You can see you can merge FCS files, LMD files here, and you'll just need to make sure that when you're merging files, you need to select this Merge FCS Files option and not one of these many other options we have for importing in data files. And once you've done that, you can just select one, several, or all the files in your folder, and you can open them, and they'll be merged and concatenated. And so when we merge our data files, several things will happen. First, we'll see that there's a new merged data file present in the data list here. If I expand this out, you'll see that it represents those 9 files that I had in my folder. And we can use this data file right away. There's no need to export it or save it. We can select it, and just like a regular data file, we can simply drag and drop this merged data onto the layout to add a new plot. And here I'm just going to go ahead and insert in a color dot plot. And I'll click OK. And what we can see is that now we have all the events from each of our merged data files all in this single plot. And just like any other plot here, we can treat it, we can change the parameters here. So I can change this to a different side scatter or one of my fluorescent parameters. We can move it around. So it's just a very generic plot at this point, and it just has all of our data files, all that data merged onto here. Keep in mind that the merged data files are concatenated alphabetically. This is important since the compensation from the first file will be applied to all of the following files. And what this really means is that we can't use different compensations for each file, and the compensation matrices of the remaining files are not used. So the compensation that gets applied is just going to be that from your first file, and that's going to be alphabetically the first file that gets merged. This is one major difference with the more flexible batch export tool, which allows you to select the compensation to apply to your merged data files. The merged data file will also contain 2 new parameters, the file identifier and classification identifier. You can access those just from the parameter list. You'll see I have a file identifier here. It's just a new parameter that was added. The values for the file identifier and the classification identifier are essentially used to discriminate between the merged data files, with the file identifier being used for 1- and 2-dimensional plots and the classification identifier used when working with heat maps. So what we're going to do here is we're just going to add in some more plots, and then for those plots we're going to use that file identifier, and I'm going to show you how you can use it to separate out the different merged data on your single plot. So I'm just going to move this plot down here a little bit, and we're just going to drag and drop in some more plots. So here I'm going to add in a histogram. So I'll go ahead and select histogram, and we'll just move this up to the top left. You'll see it's a standard histogram. I have my count, and then on my x-axis I'm going to change to my file identifier parameter. I'll just move this plot down over here to the left. And you can see that I have these 9 essentially little columns. Each of these represent a different data file. And this file identifier is going to be numeric. So I have my 1, 2, 3, 4, 5, 6, 7, 8, 9 files here. I can have my 1D plots. I can also just enter in a 2D plot, and we can view that file identifier as well. So here I'll just add in a density plot and click OK. And we'll look at our file identifier on the x-axis here, and you'll see it'll appear just like that histogram. But since I have a density plot and we're looking at a second parameter, we're going to see the data for that parameter here on the y-axis. And so I'll just change that from something aside from scatter. So let's look at one of our stain parameters like FTC here. By using the file identifier, we can see that each of these data files is really separate on those plots. And we can treat these different files just like we would any other data on a plot. So we can gate on these individual files. We can get statistics from those gates or create drill down plots based on those gates, which represent different files. We can use markers and gates to look at a specific file or multiple files all on the same plot. And by doing so, we can quickly compare any parameter between files. To show you how this works, let's just draw in some markers and plots on our gates. So we'll start with my histogram here. I'll just come up to my gating tab, and I will just draw in a marker. and I'll just cover these two data files right here. And I'm just going to convert this marker into a gate by just right-clicking on my plot and converting and linking that. And we'll just leave it as gate 1. And what I can do is I can apply this gate to my plot down here. So I'll just click on that plot, and from the gating tab, I can just select gate 1. And now we're looking at the data from these two files. And you'll see as I update the position of this marker, the data on that plot is going to update. And so I can capture data from a single file, from multiple files, or the entire data set here. And this will really allow me, just by moving this marker around, to compare my different samples. I can draw on gates, I can look at different parameters here. It allows us for a very flexible analysis. And this is something that we can also do on these two-dimensional plots with just our standard gates. So I can add in a rectangular plot here, or a gate, just by clicking on that rectangle in the gating tab. And I can gate a single file here just by drawing a gate around that one population there. And I'll click OK to keep that gate there. And then I can just drag and drop this and create a drill down plot. And then we can look at whatever parameters we want for this one specific file here. So I'm just going to look at those same parameters I had on my left, my 45, and then a scatter parameter here. And again, as I move this gate around, you'll see that the data is going to update. But what I can also do here is I can expand this out with my two-dimensional plots, and I can even focus in not just on multiple files, but on specific aspects of a parameter on multiple files. So I can look at my CD3 moderate expressors here or low expressors just for these subsets of files here. And this is one way, again, where we can just do quick comparisons between some or all of our different merged files. So with the new Merge FCS Files option, we can also display our merged data in plate format using heat maps, even if those files were not actually acquired on a plate. FCS Express will read the merged data file and essentially create a virtual plate from the merged data and display it as a heat map. And this gives us access to all the different tools and features associated with the heat map display in FCS Express, allowing us to easily visualize our samples across multiple parameters using things like the well radius statistic or overlays or custom cover levels and schemes, just to quickly compare and contrast our different samples across those different parameters, all on that heat map. And so to add in a heat map, I'm again just going to come up to our data list, and I'm just going to drag and drop this merge file onto my layout here. And we'll just select heat map this time. And you'll see that that just gets immediately plotted. And I'm just going to expand this out so we can see our heat map a little bit closer. With heat maps, each file will be displayed as an individual well. So you'll see I have 9 wells across here. And we can use the features of the heat map to compare each of these files with each other. Here we've plotted the classification identifier down here on that x-axis versus a single parameter. And remember, with heat maps, we use the classification identifier to distinguish between the different merged files and not the file identifier, which we use for our 1- and 2-dimensional plots up top. Now on our heat maps, we can easily change the parameter to visualize just by clicking on the axis. So if I wanted to look at something aside from this time parameter here, I can just click on this axis here and I can change it to our FTSE parameter, and you'll see that the colors have changed. We can also format this heat map just like we would any other heat map, just by right-clicking on it and going to the format options. And from here, I can change multiple different aspects of my heat map here. So I can change the number of columns I wanted to display just by going to the heat map specific options up in the formatting tab. And I can come here and change the columns to, let's say, 5. So I can decrease that down. So we'll end up with 5 columns, and that'll end up with 2 rows since I have my 9 files here. And if we'll preview that, you'll see I have my 5 files here and then my 4 leftover files in that second row. I can also change the orientation of this heat map. This is something that's helpful if you have a lot of different files. You want to view them in a different format than this landscape. We can go to portrait, can preview that, and it's just going to display them row-wise and column-wise here. So I'll just go ahead and click OK. So a lot of flexible options with these heat maps. It's important to note that you can only use heat maps if you have the high content add-on enabled. If you have not purchased this add-on, go ahead and give us a call and we'll help you get set up for high content analysis in the software. And with that, that really covers the basic features of using these merged FCS files. Were there any questions on these kind of just basic set of features with the merged file, how to generate it, or anything specific that we were interested in here before I move on? Okay, so for our next example, we're going to show you how easy it is to use merge data with templates. And what we're going to do here is we're going to open up a template that I've already created to compare different samples from an antibody titration experiment. So this is something that you might do in your own lab, on a monthly basis with an antibody that you might use quite commonly. So this might be a little bit more relevant to what you're doing. So let's just go ahead and open up that layout here. So I'll go ahead and click File, and we'll just navigate to where my titrations example is. It's right here. I'll click Open. Now, this is a template, and so when we open it up, it's an unlinked layout. There's going to be no data on here. And what we're going to do is we're just going to add in our merge data, and we're just going to populate this template. You'll see how easy that is here. So again, to add in a merge data file, all we need to do is just go to this Add Data list here, and our Add Data files in our data list. And then we're just going to select the files that we'd like to merge in. We're going to select the Merge FCS Files option. It's already selected there, and I'm just going to multiple select these files and open them. And the software will just merge and concatenate these 7 files here. And then all I need to do is just simply double click on them and then let the software know that I want to replace this file on all these plots. So I just click OK. And now you'll see that I have my merge analysis here. Just a few quick clicks to incorporate that merge analysis with my template here. In this example, we are titrating an APC conjugated antibody. So you can see that's an APC here on my virtual plate. And our population of interest will be present in this monostate, which I've created on this scatter plot here. And then on this virtual plate to the right, which is gated on this monos population, we have each data file distinguished again by that classification identifier. So you can see these data files are separate in each of these wells. On the y-axis, we have our statistic showing the APC conjugated antibody. In this case, we're looking at the arithmetic mean statistic for the APC fluorescence. Again, when I say virtual plate, these data files were not collected in plate format. These data files were collected as individual tubes. And with the file merging, what we were able to do here is we were able just to quickly merge these files together and then view them on a heat map, even if they weren't collected as a plate. It's just a very quick and simple operation to view them in this format. So with heat maps, we can quickly compare our different samples. For this antibody, we can see easily here which titration the APC fluorescence starts to significantly change or increase. We can quickly pick out the dilutions we're most interested in or have the highest fluorescence. And to demonstrate just how easy it is to compare between these different samples, I'm just going to add in a histogram here. So I'm just going to drag and drop in from the data list our merge data file. We're going to select histogram. And what we're going to do is we're going to expand that out, and we're just going to look at APC here. And what I'm going to do is I'm going to insert in a well gate, and that's a feature that's specific for heat maps here. So if I right click on my heat map, I can go to create gate, and I can just insert in a well gate. And from here, if I click on one of these wells, I will have created a gate that's specific for that data file. So I'm just going to make this a nice dark blue color so we can see it on our heat map very easily. And you'll see up on the left over here, I now have a gate here that corresponds to my well gate, just this one particular data file. And if I click off of that, I've created my well gate, and I can just drag and drop it from my gate view and apply it to my histogram. And now what I can do is I can just cycle through these different files. I can just edit my well gate to look at the next file or the next file or the next file, and I can look at the fluorescence for my antibody here. So I'll just right click on this heat map. I can go and edit that well gate, and I can just move along, and you'll see that the data is updating in real time, and we can view how the fluorescence looks as my titration increases in the amount of antibody that's added. And we can look at one or multiple files just by doing a control click to add in these different files. So we can look at all of our data, or we can exclude individual files if that's just not what we want to look at. So I'll just go ahead and leave this as our highest fluorescence there. And we're just looking at that single data file here in our histogram. With heat maps, we can also change the color levels and schemes to help us better visualize the differences between our data files. So if I right-click on this histogram, we can access those options from the Format menu. And from here, I can go to my heat map, and I can go to color levels. And what I can do from here is I can select the type of color level that we're looking at here. We have a standard green-red set to default here, where the green is going to be the low value of this parameter and the red is going to be the higher value on the range of all of these different values across this data set. But I can change that to be something like a red gradient or a blue gradient or grayscale. So here I'll just show you a red gradient example. I'll click preview, and you can see now that I can view my data and they're spread across a red gradient here, and it makes that maybe a little bit easier to pick out where these transitions are in the fluorescence depending on the titration of this antibody. I can also change the color level properties, so we're looking at those colors across a fixed range, but what we can do is we can change it to look based on a percentile or a threshold. So if I want to set a particular value for my arithmetic mean for my EPC and have the color be one value below it and another value above it, so I can just kind of quickly do a yes no for am I reaching a certain fluorescence threshold. You can use that threshold option, or we can change this to like a mean plus or minus standard deviation, where the further away I am from the mean of all these values, the further number of standard deviations that value is away for this arithmetic mean, I will have a different color. So here I can just show you that. We can just change the number of standard deviations to reflect the spread of this color range here. And so you see when I move that down, it's much easier to see where this fluorescence steps up between the low and these moderate APC stained wells. And so I'll just go ahead and click OK there. One last thing I forgot to mention is that if you right-click and go to Format and go to the Overlays options here on these heat maps, you can access the statistic that you're showing along with the parameter. So rather than showing the arithmetic mean, if I wanted to show something like the median, I can select that preview and we can change that out as well. So there's a lot of flexibility with using these virtual plates, these heat maps. And overall, it's just really easy to use templates with file merging and to utilize these virtual plates in our analysis for really normal day-to-day tasks, like quickly comparing between different samples across a parameter of interest, or for things like antibody titrations, which we might do on a more regular basis. File merging is a very flexible option, and with heat maps, it makes it just much easier to pick out and compare those different types of changes with your data set. So for our last example today, we're going to show you a layout using file merging and the tSNE transformation. Merging data files is particularly useful when performing data transformations in FCS Express. When we apply transformations like tSNE or SPADE, we usually want to apply those algorithms to multiple files simultaneously. That way we don't end up with different transformation results for each file. By merging our data files, we can create data that is more representative of all the samples in our experiments, since we can include all of those samples within our same analysis. And with the Merge FCS File option, we can do this very quickly. So what I'm going to do is just open up a tSNE layout. We're going to do a tSNE example here that I've already fleshed out. I've already gone and created all the plots and merged the data. And so we'll just go and open that up right now. So we'll go to our Open button here, and then we'll come to our layout. And here it is. Here's my tSNE example. Again, all the plots are already set up. The data's already been merged. I've already performed the transformation and applied it onto the layout. For those of you who are not familiar with tSNE or t-distributed stochastic neighbor embedding, tSNE is a non-linear data reduction algorithm that takes high-dimensional data and tries to find a faithful representation of that data in 2 dimensions. Researchers often use tSNE to explore and visualize their higher dimensional data and find groups or clusters with similar features within their data set. In this layout, we have merged 5 FCS files. So you go to our data list here and you can see that we have that 5-file merge here. And we have analyzed those 5 files with tSNE. And you'll see on my transformations window off to the left, I have my tSNE for my 5 files here. And this tSNE, we have selected 6 parameters to perform that tSNE on, that tSNE transformation, and we've included only events within our cells gate. And you can see our cells gates defined here at the top left, where we are essentially just excluding out some beads and some debris and just keeping in the majority of the events that we're interested in here. If you'd like to learn more about tSNE or the different transformations that we have in FCS Express, I would encourage you to attend our transformations webinar this Friday, April 6th at 1 p.m. Eastern Standard Time. I won't be going over the specifics of tSNE here, so definitely check that webinar out if you would like to learn more. Now, with tSNE, we can use the merge file to quickly focus in on the tSNE results for one or several of our files using the file identifier parameter that was created when we merged our files. So in this layout here, we have our gate, we've created our tSNE, and then up in these top plots here, I have my tSNE results, I have my tSNE X and Y, and this is just a standard tSNE analysis where we have these 2 plots that are colored based on a specific parameter. In this case, it's CD19, and you can see that I have my CD19 high population in red and the CD low population in blue, and then I made a gate with some drill down plots below, and then we can just move this gate around to focus in on those. But what if we wanted to go to the other way around? What if we wanted to not just look at all of our results here for a specific population, but maybe some of the results, and those results could be gated on a specific population. So I showed you in that earlier density plot how we were looking at some files with that CD3 mid population. Well, we can do the same things here. We can really focus in our tSNE onto specific files and then look at those results. So in this layout, what I'm going to do is I'm just going to add in another density plot, and then we're just going to use that file identifier to essentially make a gate around the files we want to focus in on and look at the tSNE results just for those files. And so what I'll do is I'll go and insert in a density plot. So I'll just drag and drop my file here, and we're going to insert in a density plot at the bottom, and I'll expand it out because we're going to be looking at multiple files here. Make sure that fits on our layout, and we'll change this from time to the file identifier. And then we're going to change the parameter here to something a little bit more relevant to what we're looking at on these plots. This is essentially lymphocyte staining. So you can see we have CD4, 8, 3, 19. So I'll change this to one of the lymphocyte parameters here. We'll pick, in this case, we'll go with CD8. And you'll see that I have stainings that differentiated with negative and positive populations there. And what we can do here is then just make sure that this plot is gated on cells so that I'm looking at the same thing that all these other plots are looking at. So I will go to my gating tab and change this plot to be gated on cells. And that's what my tSNE was created on as well. And here we're just going to add in a gate. I'll go to my gating tab, select a rectangular gate, and I can just make a gate around this one file. We'll just call this gate 2. We'll just leave it with the generic name. And then I'm just going to apply my tSNE to this plot here. And now what I can do is I can just essentially drag and drop this gate onto one of these tSNE plots up top that contain my results. And then we'll be seeing the results only for the files that are within this new gate. So right now, for this plot on the right, we have our tSNE results for CD3. And if I just drag and drop this gate onto there, now we're looking at the tSNE results for only this particular file here. And then I can use this little gate here to drill down on individual parameters there. And then what I can do is I can take my gate that I've created, I can expand it out to look at multiple different files. I can expand it to look at a specific set of parameters, in this case the CD8 high population, or I can move that to CD8 low. I can expand that out to multiple files here. So really, once I've applied my new gate to my tSNE plot, rather than looking at the results for all the files, we're just going to be looking at the results for the files that are within this gate. And by using these gates, we can pair our parameter, like I've just done here for CD8 on my density plot, and just really focus in on any population, one or several data files. We can also easily update our analysis to include additional data. So what I'm going to do next here is I'm going to save this layout as a template. And then rather than loading it with these 5 files that we've done here, we're going to load in some additional data and perform tSNE on a larger data set, a 9-file data set. And I'm going to show you how easy it is just to add in data to your analysis once you've already set it up. So this is something that might be helpful if you're just starting to do some analysis, but you know you're going to be collecting some additional data and you're going to want to include that at a later time point. You can set up your analysis now with your initial files, save as a template, and then once you have all of your data for your experiment, you can come back and you can essentially use that template and these merged files, and you can just load it in onto your template on the fly and then do the analysis how you'd like. So I'll show you how to do that here. I'm just going to remove this gate here before I add in my template, because that gate was based off of my tSNE for my 5 files, and we're going to need to make a new tSNE for these 9 data files here. So let's go and make a template first. I'm going to go to my file menu. We're just going to go save as, and then we are going to name this my template, and we're going to save it into this folder here, and we're going to make sure that it's saved as a layout that's unlinked. So this will be my template. We'll click save, and then I'm just going to close this layout out, and we'll close out some of these other layouts that we've also made here. And then we're going to go to our File tab, and we're just going to open up that template. And you'll see it's right at the top here, My Template, in this document section. So just click on it, and it will open right up, and it is a blank template here. And so what we'll do is we're going to load in, instead of those 5 files, we're going to load in some additional data files. So it'll be from the data list going to our data set. Here are the initial 5 files that I originally loaded in to that analysis. We're going to expand this out to include these other 4 files here. And so just make sure that that Merge SCS Files option is selected. We'll hit Open File, and now my 9-file merge is loaded onto my layout here. And you can see that it's 9 files there in the corner. So one important thing to note is that the data file in the data list needs to match what is in our tSNE transformation over here. So once I've loaded this data onto this layout here, so I'm just going to click on it. I'm going to come up to my change file options, and I'm just going to change the data on all my plots here, and that's going to load this 9-file merge onto my layout. You'll see that this is for my 9 files here. So we want to make sure that this 9-file merge is going to match what's in our transformations. So if I go to my tSNE 5 transformation, you'll see this template file. It's looking for a 5-file merge, and I have a 9-file merge. So what we're going to need to do is we're going to need to add in a new 9 merged data file for our tSNE. So we're going to define that new transformation up here in our transformations window. So I'll just click on Add Transformations, select tSNE, and we're just going to call this our tSNE 9. And then you'll see here that my template file is now associated with this 9-file merge. So the data list and my transformations as template file, those are going to match. And that's what's important for making sure that this tSNE is going to be applied properly for these templates. So now that I have my data loaded in, merged and concatenated, I just need to define this tSNE. So I'm going to define it the exact same way I did for my 5. I'm going to make sure that we're only looking for our gate options on the cells population, so getting rid of the debris and the beads. I'm going to use the same parameters here. I'm just going to click on these 6 here. The tSNE is going to be automatically calculated, and I'm just going to change the sample size so that it matches what we had on the tSNE 5. And this is just going to make our tSNE look a bit better. And then we're going to just select that tSNE and apply it to our different plots. And then once I've applied it to all my plots that are showing my tSNE results, what I can do is I can just go ahead and draw back in that gate. And you'll see that I have my 9 files here. And so I'll just add in a gate, and I'll make a gate around my original 5 files. Click OK, and I can apply that to my tSNE result up top. And again, we can use this gate to focus in on whichever population we want here. Again, these colors are based on CD3. But what I can do now is I can expand this gate to incorporate my new data files. And you'll see that the tSNE results are updating a little bit. It's a lot of data, so you're not going to see major changes. But if I just move this over, you're going to see now I'm looking exclusively at my new files. And again, I can do the same things with this gate. I can look at this CD8 high population across my new files, a mix of my new and my old. I can just move this around and just kind of gate in and focus in on any population that I'd like here. And you'll see that this layout is just like the one that we had open before, but now with the 9 files loaded in and transformed with tSNE. And that whole process really only took a few seconds to do. So it's very simple to work with templates and this new file merging option here. Now, with that, we've come to the end of our examples. So I'm just going to go ahead and recap what we covered today. So I'll close out this example here. With the latest update to FCS Express 6, there are now 2 options for file merging, providing you with new and more flexible options for data analysis. With the new Merge FCS file option, we have introduced a convenient way to quickly load and visualize multiple files on a single plot. And what I've done today is show you a few different examples of how to do that. We can easily just load in things on the fly and analyze them. But we can also apply this new file merging feature to different types of use cases where it's going to be most helpful. So I showed you that titrations example where we were able to easily load in our titrations data from a template and then view that data using our new virtual plates, which allow us to display data on a heat map even though it wasn't originally collected on a plate. I also showed you how we can use the Merge FCS file option to quickly adjust the files that we include or that the data that we are viewing when performing data transformations like tSNE. We were able to use gates to focus in our analysis on different sets of data and really just go back and forth between our data, the different parameters, and our tSNE results. And it was very easy to just redo that analysis as a template with our Merge FCS file. We just simply loaded it in and moved on with our analysis. So with that, I'd like to thank everyone for coming today. If you have any questions that weren't covered in our webinar, please feel free to stick around. We'll be answering questions for the next few minutes or so. And if you don't have time to do that, you can feel free to reach out to us via email at our support@denovosoftware.com address here. To download the latest update of FCS Express, which contains the file merging feature we've covered today, you can visit our website at www.denovosoftware.com. And I'll go ahead and just show you our website here. And once you're at denovosoftware.com, you can download our newest update here, or you can just come to the download page and go to all downloads and select the installation of FCS Express you have to update to the newest version here. Also, if you're interested in attending any of our webinars, you can register for those here on the right at our homepage. The transformations webinar that I mentioned, you can just directly click on that link to register for that. That'll be on Friday. And with that, I'd like to open up the floor for questions. So thank you all for attending.